Hello everyone. So in today's session, we are going to discuss about this cuff diff result. So in this cuff diff result, you can see it is giving the expression at the CDS level, gene expression level, and the isoforms level. But out of all these files, the main file is the gene expression.diff file. Why? Because in the reference based RNA sequence, the aim is to just to check what a gene expression. So everyone should open this gene expression.diff file with the help of Excel.
Hello, ma'am.
Yes,
Ma'am, I'm from NGS batch 7. Can you share this file in that group?
I have shared this file in the group. You are not added in the group?
No, I'm there in NGS batch 7, but it is
And just
not
batch
shared there.
7. Okay, just wait. Meanwhile, others can open this gene expression file in the Excel. Delimited and make it finish.
Ma'am, should we convert that to Excel or else we should directly open?
You can convert that in the Excel because every time you cannot open, right? Because
Yes.
if you are going to edit here in this sheet, we need to save that, right?
Ma'am, how to convert that?
I will tell you, just wait. Meanwhile, let me just share this. Pritish, are you added in the drive? Do you have drive access?
I don't, I'm not aware of it.
Mm,
Ma'am, can you share it here so that I can have?
But I cannot share it in the Zoom, I guess.
Okay, okay.
The
Ma'am, are you there on any WhatsApp group, like any NGS batch 7?
mm, Actually, I'm not in the NGS 7 because I was not conducting your session. So maybe what you can do, I will share my number in the chat box. Just send me hi there.
Oh, okay, ma'am.
Rest of you guys are having this gene expression file with them.
Yes, ma'am.
Okay, fine.
Ma'am, how to open this gene expression file in Excel, ma'am? Because I'm not getting Excel in open with the.
Okay, just wait. I will open it again in front of you. Just wait. Let me share this file with everyone because
Okay.
it's not added in the group.
Okay.
Yeah.
Ma'am, the file you just shared is basically the file we got from the cuff diff expression.
Yes. Yes,
Oh,
Divyansh.
okay, ma'am.
Divyansh, you are left with the cuff diff?
No, ma'am, it's not.
Okay. It's the same file, Divyansh.
Okay, ma'am.
Pritish, please send me.
Ma'am, is this your WhatsApp number?
Yeah. Please send me fast. We have to begin with the analysis.
Okay, okay.
Hello.
Yes.
I'm also in NGS Batch 7.
Okay. So, so there are a lot of people, so I cannot send it now. But what we can do, like after this session, I will share this file with you. Okay. Meanwhile, just look at what I'm doing. Okay. And you will have the recording.
Okay.
Yeah.
Ma'am, it's showing invite option. Like I'm not
Okay.
able to
Don't
see.
worry, Pritish. I will share it in
Okay,
the
ma'am. You can
NG7.
share it now.
I
Yeah,
will
you can
ask
share it.
HR to add me in this NG7.
Okay, okay, ma'am. No problem.
Okay, so this gene expression file is the main file. So for this file, we are going to see the analysis part now. So can you see in the first A column, this is your test ID.
Yes.
So what is this test ID? Can anyone tell me? So you can see test ID is what the XLOC ID, which is generated by the cuffdiff tool itself. So if we take this XLOC ID and I try to search in any of the database, you won't be able to get any information from other database. Why? Because these IDs are generated by the cuffdiff tool itself. Okay.
Okay.
So this is of no use. So what you guys can do, just delete this. Done.
And can you just show how to open the
Yes,
curve diff
done.
in Excel?
How to open in the Excel?
Yes, ma'am, because I'm not getting that.
Okay, wait. So I will repeat it again. Just open the Excel. Please mute yourself, guys.
I need to say you need to go by 4.
Desiree,
Don't worry.
Divyansh, Prayukta, can you see?
Yes,
Hello.
ma'am.
Now, after opening the Excel, go to open. Done.
Yes, ma'am.
After open, can you see the browse option?
Yes, ma'am.
Now, select the browse option and just go inside the raw data, then the cuffdiff. And what you do, just make it to all files. And just select that gene expression file.
Yes,
Done.
Yes, ma'am.
Okay. Now, are you able to open this Excel sheet,
Yes, ma'am.
Prayukta?
Yes, ma'am.
So I will repeat, this is the main file and this is your gene ID now. Can you see? Next. Can you see this gene ID? Now again,
Yes.
in this gene ID, can you see the XLOC ID again we are getting?
Yes.
So again,
ma'am.
if I try to put this XLOC ID in any of the database, I am not going to get any information because this again cuffdiff tool is generating. Okay. So again, you have to delete this.
Okay.
So this is the now gene. Can you see?
Yes.
So this is the important column here we are getting. And you can see the NM IDs here, right? So all the transcript IDs is there in this gene column. Now, can you tell me how many transcripts are there in your file? Just give me the number.
4.
How many?
4, ma'am.
4.
4?
No. 1589.
Which, how many? Abdullah?
1519.
Correct. So
Understood.
what you have to do, just press Control Shift down arrow. It will just pull it to the end. You can see 1590 transcript is there.
Yes.
Clear?
Ma'am, I have a doubt.
Yeah.
Ma'am, NM is only the transcript one, right? NR is like non-coding on it.
Narinder will discuss each and everything. Just wait. Okay. Did you got it? Till
Yes,
here?
ma'am.
Okay. Now again, if you want to go up, just press Control Shift up arrow and it will go up. Now next in the B column, there is a locus section. What is this locus now?
Chromosome.
Yes, Jay.
Ma'am, this is the location of the specific gene on the chromosomes.
Okay. So locus is telling whether this transcript has expressed and from which chromosome it is coming, that chromosome location it is giving you. And you can see first it is giving you the chromosome 16 location, and this is your start position and this is your end position. Okay. So this is the coordinates of the transcript. Did you got it? What is the locus?
Mom, this
11,540.
Abdullah, what
Yeah,
happened?
Mom, there is the starting and the ending of the fragment for the number. For example, the first John. I mean, the localization, the locus of this gen starts for 11,554 and ending on 140.
Abdullah, you can check it from the chromosome 16. When you are downloading the chromosome 16 from the UCSC also and also from the NCBI, you will get the start and the end position of the chromosome 16. So we are discussing on the chromosome 16. There are like multiple location you will get, right? And
Yes,
that location of is of transcript, correct? So
correct.
now at home, what you can do, just open the chromosome 16 and from the NCBI, okay?
Yes.
And you will get that start and the end position there. So
Okay, Mom.
let me complete and I will take all the queries afterwards. Otherwise, like it will get confusion for other also, please.
Right.
Now let's discuss what is the sample one here. So now any idea regarding the sample one here? Any idea? Sample one, sample two?
Yes, the control and the treated one.
From
Yes.
the raw data.
Yes. So sample one, sample two is containing what here? In this control, and this is the treated one, right? So why we are
Yes.
getting in this manner? Because we want
We are
to
comparing.
check the treated expression with respect to control. So if you
Yes.
see the pipeline, so I've taken this plan, right? Treated versus control.
Yes.
That is why first they are giving you the expression of control, then they are giving you the expression for treated. You can see here in the Excel, control first, then treated. So now from this data, it is very clear our plan is what? Treated versus control. Did you got it? What it is?
Yes, Mom.
Next
Yes,
thing
ma'am.
is status. So it is very important, this one, noticed and okay. So any idea, like have you ever heard about this notice and okay, Jezreel?
No. Nothing.
Okay, let's discuss what is this notice and okay. So whenever you will get the word noticed or okay. So okay means test got successful. Okay. So that means
Okay.
it's fine. Data is good for the okay. Now, sometime you are getting the low data also. So what is that low data? So our reads are not that much good, which is aligned on our reference genome. Clear?
Yes.
And sometimes you are getting high data also. Now, what is the high data now?
Enough
So there
reads.
is much the reference genome.
Enough read.
So many reads are mapping at one position. Clear?
Yes.
Whereas no test. No test means not enough alignment is provided. Okay. We did mapping, right? But enough mapping is not done. That is why sometimes we are getting as the no test option. But when you see like in the Excel, that is the okay status. Okay status. Okay. So okay, that means what? The gene got significantly expressed in your data. And when you get other type of status, that means something get wrong and those genes are not expressed significantly in your data. Clear regarding the status? Next
Yes,
thing is
ma'am.
the value I1 and value 2. So this value 1, we are getting the expression of which sample?
Control. Control.
Correct. So we are getting the expression of the control sample. So how the genes are expressing in the control sample, that expression they are giving you. But remember that whatever the data we are getting here in the value 1 and the value 2, this is the normalized count of your control and the treated sample. So I told you this is not exactly the read count. This is the normalized count after removing all the biasness and the error in the data. So it is in the FPKM value. Got it?
Yes, ma'am.
Now, can you see currently we are getting 0 here
Mm.
and
Yes.
here status, as we can see the no test here. That means the genes are not expressing significantly. That is why we are getting the 0 expression here. Okay. Now, very important column is that is the log to fold change. What is this log to fold change? Any idea, guys?
Is it
Did you
no?
heard about this log to fold change?
Yes, ma'am.
What it is?
Ma'am, here we compare, I mean, the treated and the control and see the difference. Either it's 100% more expressed, I mean, we can say it's one-fold. And if it's just like, for example, control is 2, treated is 4. So we can say that it's one-fold or two-fold like this.
Correct, sir. Correct, sir. So whenever we are doing the differential gene expression or whenever to compare one sample with respect to another, that is what the fold change. So it is basically quantifying the difference between the sample. And why we are taking this log 2 here? Because now if I'm getting the result in the form of positive,
Yeah.
so positive means what? My gene is expressing, right? It may be the upregulated.
Upregulation.
Yes, correct. Now, what if I'm getting the negative values there?
Down regulate.
Correct. So negative means down regulated here. So upregulated and down regulated. Upregulated means process in which cell is increasing the quantity of their cellular component, whereas in the down regulation means this is the process in which a cell decrease the quantity of their cellular component. Okay, now let's discuss why they are taking the log 2 fold change. So I'm giving just the example for you guys so that you can understand what is this log 2 fold change. So for example, I'm having 3 genes with the name G1, Gene 2 and Gene 3. And I'm taking 2 samples, that is the control and treated. And now from the control sample, there are 10 reads, here 20. And for the gene 3, I'm getting 10. Whereas in the treated 20, I'm just taking an example, okay guys? So this is the reads here. Okay. Now, if I want to calculate the difference between the control and the treated, what I will do? I will take the plan as the control divided by treated, and further I will take treated divided by control. So let's calculate. Equal to, so control divided by treated, okay? Control divided by treated. Enter. So I will autofill this data. And here my plan is treated divided by control, right? So equal to
C4 divided by C4.
So this is not the fold change, okay? So can you guys tell me from this data, which gene is showing the upper regulated and which gene is showing the down regulated? Can you tell me? Any one of you?
Mom, the G3 and the G1 is up regulated.
Why, Abdul?
As
G1 and
for-
G3.
No.
Jezreel?
G1 and G3.
Ma'am, I think all the genes are upregulated.
No.
Okay.
G1 and G3.
Yeah. So I can see from you guys, guys are telling me like these genes are upregulated and the downregulated, right? But in real time, you will get the multiple data, right? 1,000 of genes you will get, correct?
Yeah.
So for the 1,000 of genes, you cannot manually tell me which gene is giving what, the upregulated and the downregulated, correct? So from this data, we cannot clearly mention which gene is expressing high and which gene is showing the less expression, correct? So that is why we are introducing here the log 2 fold change, guys. Okay.
Yes.
So if I'm taking the plan here, control versus treated, okay? So anyone of you, like, do you know the formula of this log to fold change?
Sorry, ma.
What is the formula of the log to fold change? Okay, I will tell you. Just put equal to here and put log, and in the brackets you have to give the control divided by treated. So we have already calculated, so I will select this from here and comma 2. Put the brackets, then enter, then autofill it. Did you got the formula? Equal to
Yes.
log bracket. So which is your plan, treated versus control, right? So I will just select this, comma 2, enter. So this is how the tool is generating the log to fold change values. Now, can you tell me again for this data, which gene is showing up regulated and the down regulated? Now, can you tell me?
Ma'am,
Ma'am,
G2
the
is
gene one
Positive
is
one is upregulated
upregulated.
and the minus one, negative one, I mean.
Correct, Abdullah. Okay, so there are standard cutoff for the log to fold change. So if you are getting log to fold change value that is equal, greater than equal to 1, that will indicate what? It is the upregulated. If it is less than equal to -1, that is down regulated. Next, if it is equal to 0, that means gene is showing no expression, guys. Just mute yourself, guys. Okay. 0 means no expression, okay?
Yes, ma'am.
Now, why we are performing the log to fold change value? Because this log, from the log to fold change value, you are getting the result in the form of positive values and the minus value. Whereas this positive will indicate the upregulated genes and minus will indicate your downregulated genes. So now there are two plans. Can you see? This is the control versus treated plan and this is treated versus control. Okay. Now, for particular gene 1, you have to tell me which gene is showing higher expression in which plan, guys.
Ma'am, gene 1 in treated versus control.
Yeah, correct. Who is this?
Prayukta, ma'am.
Prayukta, nice. So you can see you have to be very careful with your biological analysis plan. Why? You can see it is very clearly mentioned here for the G1. If my plan is treated versus control, it is giving you the upregulated genes, which is indicated with the 1. And also from the count of the reads, you can see this is 20 here, correct? But if I take the plan that is the control versus treated, it is showing the opposite to that. Can you see that? It is showing down regulated. And
Yes.
from this read also, you can see it is showing less reads there in the control. Okay. So
Yes,
is it clear? The log to fold change.
Yes.
Yes, ma'am.
Any doubt?
Ma'am, biological plan for control, treated control. We
Abdul,
make this.
Abdul,
Yes.
see, whenever you are performing the procedure in the wet lab also, you have to take some things control, correct? So for example, if I'm dealing with the cancer patient data, okay,
Yes.
and I'm working on the tumor there, so how I will come to know whether that patient is having tumor or not? So I have to take the as a control of the human reference genome there, right? So that I can compare my cancer patient data with the normal human genome, right? If I will compare that data with the normal one, I will come to know which base is mutating in the cancer patient data, correct? So
Correct, ma'am.
that is why we are taking something the standard here, okay? Whenever we are doing anything in the experiment also. Got it?
Yes, ma'am.
Now, next thing is the test stat here. So this is nothing. This is just a statistical score which is given by the CuffDiff tool itself. So you don't have to worry with this statistical score. It is just a mathematical, okay, part. So what you have to do, just delete this from your file. Done?
Yes.
Now, what is this p-value? Can anyone tell me what is this p-value?
The risk value. do you mean?
Risk value?
The p-value is just telling us if there is a significant difference in the expression.
Yes, sir. Correct. You are right. So basically, p-value stand for probability of the error value. So it is will tell you how significantly our gene is expressing in your data.
Ma'am, this value it's our we fix it before the check to run this analysis. I mean,
Yes, sir.
it's known by 0.05.
Yes. We will discuss.
Yes.
Just wait. So p-value. A smaller p-value will suggest a lower probability of observing the observed difference by chance. So this generally indicate a strong evidence for the differential expression. So whenever you are dealing with the p-value, so there is a standard cutoff for this p-value. That is, it should be below less than equal to 0.05. So this is the cutoff for the p-value. Remember that it is always the same. So whenever the p-value is below the 0.05, that means our gene is significantly expressing in our data, guys. Clear?
Yes.
So the gene can be expressed in negative as well, the p-value. Or
No, p-value does not have negative.
Yes, ma'am. See, Prayukta, we will see what whenever we are dealing with the will provide the filter there in the Excel sheet. Just wait, okay?
okay,
You will
okay,
see the result, okay?
okay.
So for your answer, I would suggest like the genes will express, but it will give you the less expression. So it will be the down regulated genes, okay?
Okay, ma'am.
Yeah. So next is very important again, the Q value. Q value in the CuffDiff result is a corrected p-value that
Adjusted.
is taking into the account of the multiple testing problem. So when we are analyzing many genes or isoform, there are some chances you might get the false positives, right? So Q value is what?
Adjusted p-value.
Correct, sir. Who is this?
Yes.
Jezreel. Nice, Jezreel. So Q value is the adjusted p value. So adjusted p value to control the false discovery rate. So we are also dealing with the FDR. So we call it as the FDR also. So it is a proportion of the false positive among all the genes and the isoform, which is identified as a differential express, and lower FDR indicate a lower number of false positive. Clear P value and the Q value. So
Are there is there any relationship between the P and the Q?
yes, Abdullah. See, before the Q value, the P value is calculated. So there are some chances you might get the error. Why? Because we are providing here the 5% of the error, right? If you remember in the notepad,
Yes, ma'am.
I have written 0.05.
Yes, ma'am. Yes.
So what is this? I am applying the 5% of error in my data. Okay.
Yes, ma'am.
So
Yes, ma'am.
there are some chances you might get the false positive result. So that false positive result, it is being corrected with the help of Q value. Abdullah?
Oh, yeah. Like double check.
Yes, correct. Okay.
That's my right.
Now, next thing is the significant. So it is of no role, like significant no means it is showing no expression. Yes means it is showing yes expression. But you can delete this significant. It is not important for our data. We are going to deal with the P value and the log2 fold change value. So these two are important. Clear everything. And this is your main file, guys. Just save this in the Excel format.
Ma'am, sorry. Is it always easy to explain the result like you do here, or something? Sometimes it will be so difficult.
No. It is as like the file which you are looking at right now in my system, the same file you will get for the other experiment also, sir.
Okay, ma'am.
Yeah.
Ma'am, I have one question.
Yes. Who is this?
Imran.
Yes, Imran.
Ma'am, I think usually in our experiments, we get the control versus treated. I mean, the log2 fold change values. Maybe I didn't see in any experiment where we have compared, I mean, treated with control.
No, it is depending on which data you are dealing, sir.
Okay.
See, if
okay.
You are dealing with the de novo data, okay?
Okay.
So that time the result will be different and the pipeline will be different for that.
Okay.
So this is basically you will get only when you are isolating the RNA, sir.
Okay.
And also when you are dealing with the transcript only. Okay. What if I want to deal with the cancer patient? That time I am not going to analyze this type of data, sir. Data will be different for that DNA sequencing. Application will be different, sir.
Okay.
Ma'am, the
All right.
example black one, is it the same or what we should do?
What? Abdullah?
For the black one, the BRA1.
Yes, BRCA1 that is present in the human.
Biological plane, I mean, it's control treated or treated control?
No, sir. Whenever like this BRCA1 gene is what? It is causing cancer in the human, okay? So, so if you want to see the mutation there, I am not going to follow this pipeline, sir. This is not, we are going to run this. So there is a different pipeline for that, sir.
Okay.
So there are different application in the NGS, sir. Okay. So basically,
Sir,
like
I think it depends on the analysis that we are
yes,
doing.
sir. Yes,
If
sir.
what we are doing is genomics or variant calling, is completely different thing.
Yes,
But if you still want to look at the RNA-seq gene expression,
yeah,
we can
so
still
that time,
use the same pipeline. But if we are
correct.
using genomics, it's going to be completely different pipeline.
Exactly, sir.
Yes.
You got it, Abdullah?
Yes, ma'am.
Because whenever you want to call the variant, you are going to deal with the another application, not this. So it is basically we are dealing with the reference-based RNA sequencing. So Every time if you are dealing with the reference space, so same data you will get that. Same data. So nothing difference is there.
Yes, ma'am. Yes.
Clear every each and everything it is clear in this Excel sheet now?
Yes.
Okay. Now let's see further.
I have a question.
Jessel, yeah.
Yes, please. Based on this Excel data sheet,
Yes, Jessel.
do you think there's still need for control for the quality of this data based on this no test and okay,
Yes, yes, yes.
everything will be included in the analysis?
No,
don't you
sir.
think?
No, sir, Jessel. We are
Okay.
going to apply the filter further. I was
Okay,
just
okay,
explaining
okay.
because
Okay.
people are very new to this analysis. So I have to discuss what is this notice and okay before putting the
Okay.
filter. Okay, Jessel?
Okay.
Just
Okay,
wait,
yes.
we will discuss further.
All right, thank you.
Okay. Now, in the post analysis, our first step is to sort the significant gene. So what is a sorting of significant genes?
It's a kind of arranging the gene based on the p-value, on the adjusted p-value,
Yes,
so that
correct.
we can either have in ascending or descending order based on
Yes,
what we
sir.
expect to
Yes,
go.
sir. So this is your data which you got from the cuff diff. So it is not possible like each and every transcript is expressing in my file, right?
Yes,
So as you can see, there are a lot of transcript. So we are going to provide the filter here so that we can deal with only with the significant gene, guys. Okay.
Yes.
Then next step, after doing the significant of genes, if I want to know like the gene annotation, so you can see in this file, if I ask you on this transcript which gene is present, so you can see in the Excel sheet there is no column for the gene name. Can you see that, guys?
Yes.
Yes, ma'am.
All that annotation we are going to perform with the help of UniProt database.
Okay.
Further, if I ask you what is the biological, molecular, or maybe cellular function of this transcript, so you can see there is no function here in this data, correct? So that also we are going to see in this enrichment. That is the functional enrichment. So in the functional enrichment, we will discuss which gene is showing higher expression in which type of function. So that we will see in this enrichment. Then pathway enrichment we will see with the help of KEGG database. And functional enrichment we will see with the help of Panther database. Next, what is this network analysis, Jezreel?
Yes, there ought to be the connection between the genes that are expressed.
Yes, correct. So network analysis means how genes are interacting with each other so that we are going to see with the help of string database. And maybe we will use the cytoscape for that. And this is our plot is nothing. This is something additional I have put in the script. If time allows, I will tell you how to run the R Studio and the R programming. So with the help of the R programming, I will tell you how to create this volcano plot and the heat map plot. But till this step, all the data will be done in the analysis. Clear? Clear, guys?
Yes, I wish that the art plot is also included.
Jezreel, we are left only with the 4 days, and in the workshop and also in the our session, our plot is not included. But sometime what happen, people are very smart. They pick up very quickly, and I am dealing with the R plot there. So if I see like in coming next 4 days, if the session is running smoothly, so I will plan this R plot with you guys also.
I hope so.
Yeah. So is it clear what type of analysis we are going to perform?
Yes.
Okay, let's see now in this. Now, can you tell me at 6th row, what is this indicating? There are 3 transcript. Can you see that?
Yes.
What is this 3 transcript is indicating here?
So we have three genes to code for one protein.
Who is this? Abdullah?
Yes, ma'am.
Okay, Jezreel, what it is?
Yeah. Oh, I'm thinking it's multiple read. I don't know.
Okay, so this is basically the isoform you can see, as Abdullah said.
Okay,
So it
okay.
is present on the chromosome 16, as you can see.
Okay.
So never keep this type of data in your Excel.
Wow.
We want to see whether these isoforms are coming from same gene or different gene. Okay, so what you guys have to do in real time, you have to just copy this NMID, go to NCBI.
Okay.
Likewise, I will just create the duplicates. So because you can see there are total 3 transcripts. So one by one, what you guys have to do, just copy each one of this transcript and put that in your NCBI. So we will see whether this transcript is coming from same gene or different gene, guys. So now let's see. What is the gene here?
It's
The same.
gene expression.
It's the same. No, it's not open yet.
Jezreel. This is the same. Can you see?
Yes. Yeah, it's the same.
And also for
It's
the
the same.
third transcript, you can see MPG, correct?
Yes. Yes. The same.
So I have already found for each and every isoform, this data is coming from same gene, guys.
Yes.
Okay. But in real time, if you are getting the transcript from the different genes, so that time what you have to do, I will tell you. Just copy this data. Okay. Just Control Shift down and paste here. If your isoforms are from different genes, remember that. Now, one by one, what I have to do, I will just remove this ID and I will keep this. Again, again just scroll up, again copy this and Control Shift down. Now here just keep this ID, okay? Just remove this. Okay. Did you got it? What I did here?
Yes.
Like
Yes.
you splice the jam or three jam.
Exactly. We have split that into the different rows because they are coming from different genes, right?
But if they coming from the same gene, we keep it as it is.
Yeah, so I will tell you how to deal with that also. But in real time, you have to do. So it will take a lot of time because manually we have to do this thing. Okay. So for now, I will just delete this. So what about others? Did you got this?
Yeah. Yeah.
Okay. Now, if it is coming from same gene, what we have to do, simply copy this entire data and paste that in another sheet. I will just delete this. Now it is coming from same genes, guys. So it
Yes.
is of no use of having this multiple transcript, correct? So
Yes.
what you have to do, just copy that, go to data. There is an option sort, or maybe you can use this text to
Also, yes, just the column.
delimited. Next, in the others, put here comma. It will separate every transcript. Can you see that?
Please, could you repeat, Mad, please?
Yes, Abdullah. You are on the same page, guys, everyone. Did you
Yes.
copied?
Yes, ma'am.
Now, just copy the entire data.
Yes.
Go to data option.
Yeah.
There is an option text to column. Opened.
Yes, ma'am.
Now delimited. Then next. And there is an option tab, semicolon, comma, space. Just select the tab and other. In the other, put the comma there. And do it next.
Yes, ma'am. Perfect.
Then finish. Done? Done?
Done, ma'am.
Yes.
Now, what you have to do, just copy the entire data again from here and go to main file, that is the gene expression file, and you have to paste here. Now you can see it is replaced. Can you see that?
Yes, ma'am.
What about others? Divya and Shubham, you are not responding. Divya, Amit, any doubt? Are you comfortable till here, guys? Done? Okay, so we'll end here and I will send you this recording. Just look at again this today's session, and further in the tomorrow session we are going to do the annotation part, and we will sort and we will see how much genes is showing significantly expressing in our data and which gene is upregulated and the downregulated. That we will see in the tomorrow session, guys. Make sure you save this file in the Excel format. Okay.
Ma'am, after
Okay.
going to data, I'm unable to separate the columns.
Can you just share your screen, ma'am?